All right, so we're going to talk a little bit about media here, culture media, as well as some aseptic techniques on how to inoculate these types of media to where we only have the one bacteria that's present. Now, we do see that media is going to be a type, when we talk about culture media, this is going to be a type of substance that's going to enhance the growth of bacteria by supplying them nutrients. We see that it can come in two forms. It can be in a liquid form, which we call a broth. All right, and so in this case, we have a broth. See, it's in a liquid form. Another one, here's a broth that's in a liquid form. Okay, we also can have an agar, which is more of a solid or semi-solid form. A lot of times agars are going to come in plate formation. So we can see we have different types of plates based on the agar that's present. We can also put the agar into test tubes and create what we call a slant. What they do is they pour the agar in here when it is hot, and before it solidifies, they put it at a slant. And so you can see that there is a slant here. We have one also here where it's at a slant. We also see they can put it into a test tube where it's a solid, and this is called a deep. All right, and so we can see that it's a deep in this case. Now, sometimes when we look at these, some of these plates or these agars or broths are going to be what we consider a non-selective. A non-selective type of agar is going to allow pretty much anything to grow on it. Okay, and so this would be like nutrient agar, like we see here. This may also be like a nutrient type broth that's going to allow, where's my broth? A nutrient type broth that's going to allow a lot of things to grow in it. Now, sometimes bacteria is just real picky. They can't necessarily grow on something like this, just a nutrient agar. And in that case, we would supply it with what it would want or what it needs. In this case, this is a blood agar, a sheep blood agar. This has blood added to it. And this is going to be for those bacteria who really enjoy blood, like hemolytic bacteria. Okay, they like to lyse the blood cells and get the components out of them. And so because of that, this would be one that they could grow on. Sometimes these media are what we consider selective. These selective media will sometimes be used in order to select against one type of bacteria, but allow another to grow. When we look at McConkey's agar, this type of agar is going to allow for gram-negative bacteria to grow on it, but not gram-positive. And so because of that, it's going to pick what gets to grow on it. All right, sometimes we might add antibiotics to a bacterial or to the plate, and that would also hinder some bacteria from growing. Other types of media cultures are going to be differential. This is going to be where when stuff does grow on it, that's going to appear different on the plate. McConkey's is another one of those where we see that it's going to allow lactose fermenters to grow differently than non-lactose fermenters, the ones who can use lactose sugar versus those who cannot, and they're going to appear different on the plate as they grow. All right, and so we see that sometimes those are what we would see with some of these agars, is they're not only just selective, they're also differential in the sense that they allow for differences to be seen as the organisms grow. All right, so when we look at these different cultures or different types of media, there's different ways that we would need to be able to inoculate them. We're going to, on our plates, do the streak method on these plates. Unlike what we saw in the spread plate method where we wanted to cover the whole thing with the bacteria to create a lawn, in these particular cases, when we do a streak plate, we want to actually get individual colonies. All right, this allows us to look at the colony growth where we can identify what the colonies look like, if they're domed, if they're symmetrical, if they're irregular on the sides, if they're sticky. It allows us to get a lot of information just by looking at how they're growing on the actual plate. All right, so we're going to use a streak method here for these. When we're talking about our broths or even our agars that are in the test tubes, these are going to be utilized in a little different way, and we're going to use either needles or an inoculating loop to inoculate them. And a lot of these are going to be used for other tests, specifically for gram-negative bacteria, which we'll talk a little bit more in later videos. So let's go ahead and look at our streak technique. Now, when we look at a streak technique, we're going to take our kind of agar plate and we're going to put it into these quadrants. These quadrants are going to then be broken up, and we're going to inoculate them in a little different way using this method. We want to make sure that we sterilize our loop in between each of these so that we can isolate the colonies. And so that's what we're going to be looking at for our streak plate inoculation. All right, so to start off with our streak plate inoculation, we're going to use a sterile cotton tipped applicator, just like we saw with the case with our spread plate, but we're going to use it a little bit different. So we're going to take that out. We're going to use again, put it in here where we're going to collect the bacteria. We're going to get excess off. Okay. And what we're going to do is we're going to take our plate and we are going to put a line, just a line across there. Okay, that's all we're going to do with that. And then we're going to get rid of our applicator. We're going to then go through the process of sterilizing our loop. So our loop is going to be sterilized, and we are going to run it across that line that we just put in there. And we're going to do a zigzag type motion across there in order to spread that bacteria out. All right, and so I'm going to pick, once we see that it is cooled off, we're going to take this and we're going to go back and forth in a zigzag pattern across that line. Okay, now I'm going to go ahead and sterilize my loop, because what I'm wanting to do is I want to pull some bacteria from the previous area, but I want to start limiting the amount of growth in order to make sure that we're going to get some single colonies that are present. Okay, so again, I'm going to go by that zigzag area and I'm going to pull from the edge of that, and I'm going to zigzag back and forth down this way. Okay, and again, I'm going to sterilize my loop. Okay, so this is then quadrant 2. I'm going to then go through quadrant 3, again pulling from number 2 into 3, but only pulling hopefully a few of the bacteria. The whole point is if we can get an isolated bacteria to go through the process of growing, that's going to be that isolated colony. All right, so again, I'm going to go back and forth in that area. And then I'm going to do my 4th quadrant where I'm going to go through what's left into the middle. Now I want to make sure that I don't draw into any of the other 3 areas that I've already pulled, in the sense of going back into it. We want to enter into 3, pull it into 4, but we don't want any of our streaks to go from 4 into 2 or 1. All right, we want to make sure that we are separating those, okay? And so I'm going to come over from 3, and I'm going to go back and forth into the space that's available, okay? And so that's my inoculation there, okay, with my streak plate. And I'm going to have you take a closer look in a minute, okay? I sterilized my needle or my inoculating loop, put it here, but remember, we always have to label our plates. I'm going to put my initial, and I know that in this case, it's E. coli. And put it off to the side, all right? So now we're going to take a look at doing the same thing on our sheep auger. All right, so now we're going to do the streak plate where we're going to be putting it into our blood auger. All right, so I have a blood auger plate, and I'm going to utilize again a cotton tip applicator, and I'm going to collect some E. coli on it, making sure that I get the excessive fluid off by pushing it to the side of the inside of the container. All right, once we do that, we're going to take that applicator and we are going to do one line across the auger, okay, allowing that line to be present. It's not like what we saw with the streak plate where we went back and forth all the way down the plate. It was just in one area. Now I'm going to take my sterilized loop and I'm going to make a zigzag pattern across that line. All right, then I'm going to sterilize my needle again, allowing it to fully cool before I go and inoculate again. When I inoculate now, the second time, I'm going to pull from that first quadrant, that first zigzag pattern that I made, and I'm going to make a second zigzag pattern into quadrant number 2. So I'm going to pull from 1, do the zigzag pattern into quadrant 2. Once I've done that, I'm going to then re-sterilize my loop. Okay, so I have to re-sterilize between each of these quadrants. This time I'm going to pull from quadrant 2 into quadrant number 3. So I'm going to pull from 2, okay, you can see the little zigzag there, and I'm going to zigzag into quadrant 3. Again, sterilizing my needle. After I sterilize my needle and it has fully cooled, I'm then going to go from quadrant 3 into the last area available on the plate, which is quadrant 4. Being careful not to go back into quadrant 1 or 2, because again, we're trying to isolate those colonies. All right, so you see the whole plate's not covered like we had before, but we do see that we have these zigzag streaks that are present. Again, we need to make sure that we label our Petri dishes. We want to put our initials, and then in this case, we know that we're dealing with E. coli, so we're going to label it as E. coli. Now, in this particular case with the McConkeys, I don't really need an isolated colony here. So what I'm going to do is I'm going to end up just doing a zigzag pattern type of inoculation. Again, though, I want to use a sterile type of cotton-tipped swab. I'm going to still use my broth with my E. coli that I have in here. Again, I'm going to go through the process of getting excess fluid off. Lid on. Okay, and in this case, I'm just going to run it back and forth in a zigzag pattern, okay? Because it's the only thing that I'm growing on this particular plate. And this, again, is one of those selective differential. It's gram-negative, so it should grow. And then we're going to see if it can actually do lactose fermentation. Now, when we're looking at our broths and our different types of slants and stuff we have here, a lot of times we're going to pull bacteria off of a plate instead of pulling it out of what we already had here in a broth. Okay, so with this, again, I have an E. coli plate that's here, and I'm going to use this. Now I'm going to do the broths first. So I have 2 broths here. These broths are a little bit different. This broth right here is a MRVP broth. This is going to be used for the methylene red and the VP test when we talk about a gram-negative type of bacteria. All right, and so we need to inoculate this so that we can run both of those tests with this particular media that's here. Now, this is just kind of a nutrient broth that's here, and we're gonna add other chemicals to actually see the test. But in this case, we are going to go through that process. Now, we are gonna use our loop. We need to sterilize our loop, okay? Again, we don't want to lay any of our lids on our surfaces, okay, because that's where contamination may take place. And so I'm going to use my pinky here, and I'm going to pull the lid off, and I'm going to hold it. I'm also then going to take my plate, and I'm going to just tilt it up. I'm going to grab some of the bacteria, and then again, I'm going to place it in here and just move it around. This allows the bacteria to come off my loop, okay, and into the container. Okay, now again, I want to sterilize my loop after I'm done with that. Okay, again, putting the lid back on, I want to tighten it all the way and then unscrew it 1/2 of a screw in order to make sure that the gas can be released. So in this case, I'm going to again put my initials and what it is. Okay. All right, the next broth I'm going to look at here is a phenol red broth. This is going to be looking specifically at sugar type of fermentation. So it has sugar. The color may change based on if it can ferment the glucose that's in here. You'll also notice there's a little tube in there. You can kind of hear it. That's a Durham tube. Inside there, there's no actual gas. However, if this particular bacteria, when it's growing in here, produces gas, it will collect in that tube. So this is going to allow us to see 2 different things. We're going to be able to see if a color change takes place as well as if there's gas in the tube. Again, we're going to do the same thing. We're going to sterilize our loop. Okay, we're going to end up collecting some of the bacteria off our plate and adding it into the broth. So in this case, we're going to pull the lid up, collect some of our bacteria, and then we're going to place it in there. Okay, and sterilize our loop. All the way on, and then half a turn to open it up. Again, making sure we label our tube. All right, these next three that we're looking at, these are going to be dealing more with a needle. And so instead of using inoculating loop, we're going to use a needle. It's very similar to our loop, except for it's just a straight line. We're gonna collect a little bit of bacteria and then we're gonna place it into the agar. So again, I'm going to sterilize my needle. In this first case, we're going to look at this Sim media. This media is a deep, okay, so it's not at a slant here. And this is gonna allow us to see if our bacteria has motility, movement. Also, if a color change takes place where it turns black, it's gonna also let us know if it is a hydrogen sulfide producer, okay? So we're gonna take this and we are going to unscrew our lid here and get this all ready to go. Lid up, and I'm going to collect just a little bit of the bacteria on my loop. And in this case, what we're going to do is we're going to take this and we're going to go straight down into the media, okay? And then we're going to pull it straight back out, okay? That creates a deep, okay? We're also going to scratch the very top of the media just to get the excess bacteria off, okay? And then I'm going to go through the process of sterilizing my needle. Okay, again, putting the lid on, taking it half a screw back so that gas, if it is produced, can be released. Labeling my tube. Okay. Now, these next two are slants, and we're going to inoculate them the same way, except for that they have different components. This is a triple sugar iron agar. This is going to allow us to see if when oxygen is present versus when it's not present, because oxygen is not present down here in the deep part, but on the surface, if it's going to utilize the sugar in a different way. So again, we'll look for color change, as well as if there's going to be gas production, because if gas is produced, it'll pull the agar off of the base of the tube. Okay, so again, we're going to use our needle, sterilize it. Okay, we're going to do the same thing we did with the deep, in the sense that we're going to collect a little bit of the bacteria onto my needle. And I'm gonna have to hold it a little bit at an angle here because I'm gonna go down into the deep. Okay, so I'm gonna go down in here, straight down. Then I'm gonna come back and I'm gonna scratch the surface, okay, as I come up. And then I'm going to sterilize my needle. Again, lid on, half a turn, and my initials and what I have, okay? And last but not least, we're going to end up doing the Simmons citrate agar, and this, we're going to see if it can utilize the citrate that's in here in that citric acid cycle known as the Krebs cycle. And so we're going to look again for a color change. We're going to also do the deep like we did before, and then scratch the surface. So again, we're going to sterilize our loop. We're going to take our lid off. Gonna collect a little bit of bacteria, and we're gonna go into the deep and come and scratch the surface. Again, sterilizing the loop when we are done. Okay, so these processes, again, lid on, half turn, put our name on it. Okay, now these are going to go into the incubator. Some of them have to be incubated longer than others. You have to pay attention to those times. But they're going to go in the incubator, and this is going to allow us to then do further tests, especially when we look at certain things like our gram-negative bacteria. But the whole point in this part of the lab was just showing you how you can inoculate different types of cultured media, whether it's a plate, or if it's going to be a broth, or if we look at here being a deep or a slant. All right, and so this allows us to see that there are different ways that we can inoculate this type of material in the lab. If you have any questions, please let me know.